In this final video on stem cells, I want to take you through a story where people used microarrays to look at gene expression of various stem cell types and discuss the state of the art for stem cell therapies and how stem cell therapies might contribute to gene therapies, and also discuss some of the issues associated with turning this promise into a reality. The first study I wanted to talk about with the microarray expression data is from 2002, and this group of experiments did microarrays on a number of types of cells. They did hematopoietic stem cells of from both human and mice. They did neural stem cells. They did embryonic stem cells, and their goal was to compare genes that were upregulated in each of these cases to find genes that might be responsible for stem cell-like characters, the ability to replicate a lot, the ability to produce a large number of cell types. And when they looked at these studies, they're looking at the intersection of genes that were upregulated. And the first group found 283 genes that were upregulated in all of these three cell types, and considered that genes that were responsible for stem cell properties. Another group here at Harvard, same time, very similar experiment, same basic cell types, embryonic stem cells, neural stem cells, and hematopoietic stem cells. Do experiments on Affymetrix chips, find genes that are upregulated under those conditions. Did the same sort of analysis where they're looking at it, and again, about 230 genes that are upregulated. And a third study did that as well. And then a fourth paper came out to compare results from those two. The original two are blue and green in the third study. And despite being extremely similar experiments and finding lots of genes regulated, they only found 6 genes that were overlapped between the two studies. And then when compared to a third data set, the rather embarrassing result that only a single gene is upregulated in all 3 of those. So this really caused into question the materials that were being studied and the methods that were being used. And this certainly put an end to this type of experiment. It's this type of experiments have not been repeated. This was so embarrassing. But what people just realized was that the details of the cells that are being originally used, how they're being handled, how they're being grown, matter a great deal, and that there is going to also be a lot of heterogeneity. Nobody's doubting that these weren't actually embryonic stem cells or neural stem cells, but that there's a great degree of heterogeneity and perhaps multiple ways to achieve these states. Other things that were learned from this study were that the platforms used, different variations of chips were being used, and so that people had to become very careful about comparing results from different platforms. As a scientist or engineer, that's disappointing, I think, to all of us inherently. Why should it matter which chip or which platform you're using? You expect to see the same result, but this study showed clearly that that is not going to be the case. Like I said, these studies have not been pursued much, but the closest studies that have come to this are comparisons of embryonic stem cells and induced pluripotent stem cells. And there's at least 3% difference. 3% of genes are differentially regulated in those two cases, indicating that there are very likely some differences. Whether it's important for treatments or not yet, we don't know, but there are some very real differences between those different methods of generating pluripotent cells. So I have 2 slides here to give the overview of therapies, and despite all the promise, the punchline is that there are relatively few treatments that yet take a really great advantage of stem cells. But in thinking about how stem cells can treat disease, it's thought, by stem cell transplants or transplants of differentiated tissue. Definitely neural stem cells and repair of neurons with their central nervous system, spinal cord, is certainly at the forefront. Another disease that people would love to be able to use stem cells for is diabetes. The regeneration of cells that would release insulin is a major experimental focus, and lots of people are working on those. No great results to date, if you think about what diseases in principle could be treated by research, stem cell research, nearly every every disease is potentially treated by stem cells, even broken bones, infections, congenital defects, it's all possible. The stem cells therapies that are currently available, we'll show one that is clearly supported by evidence as the best possible treatment and is used quite widely. There are many clinical trials going on. Mostly, there are a lot of phase 2 trials going on currently, 1 or 2 phase 3s. So the promise is still really to come. As we'll see, the therapy that we're using is a type of bone marrow transplantation rather than transplanting the entire bone marrow. We'll separate out the stem cells and just put those back into the new patient. These will then go to the bone marrow and replenish all the cells that hematopoietic stem cells can develop, so you'll reconstitute both the red and white blood cell systems, which are then used for proper maintenance of tissues and infection status. In terms of talking about why it takes so long to create stem cell therapies, you have the problem of differentiation of stem cells into the type of cell type that you want to create. We saw that you get quite heterogeneous results in most cases. Once you've got a therapy that you think is working, they have to be tested not just in lab dishes but in animals, and usually multiple animals, because sometimes things might work in mouse but not in a rat, or not in a dog, and maybe not in humans. So that is certainly a big time sink in terms of validating treatments. There's that great concern about stem cells being immortalized and potential of creating tumors. Stem cells can definitely form a type of tumor called a teratoma, which is a tumor with all kinds of cell types inside. And for the iPS cells, we're even putting in genes that are known to be oncogenes to help get that immortalized state. If we're putting in embryonic stem cells from a fertilized egg, that's a different source. Then you will have immune rejection of non-self stem cells that are generated by somatic cell nuclear transfer of one of your own adult cells, or iPS cells. Can in principle be perfect genetic matches, but immune responses are not expected. However, cancers are at one point come from yourself, and they can clearly generate immune responses. So it's not clear. And then finally, as part of any FDA-approved therapy, the production methods are strictly regulated. As in terms of quality control, we want to make sure that each batch of stem cells behaves the same, and that has been a very challenging aspect of actually deploying these therapies. So we will, after the quick talk on the approved stem cell treatments, we'll have to focus mostly on the potential of stem cells for other treatments. And the type of approved stem cell therapy, which is much better than any other form of therapy, is hematopoietic stem cell transplantation in severe combined immunodeficiency disease, which can result from any of 10 mutations. Transplant of hematopoietic stem cells can lead to high survival rates over long periods of time. These are the best that have obtained for any treatment and are highly reproducible. The survival rate changes based on how good a match genetically the stem cells are from donor to host. And so despite being the best available treatment, people are still working on improving that using other methods. But here's a case where stem cells are the best treatment available that has been validated in clinical trial. Some of the hopes for stem cell therapy, for instance, in cancer treatment, is in the case where someone has to have high-dose chemotherapy or radiotherapy, which would wipe out the person's entire blood, the immune system, and the red blood cell system, is to go ahead and deliver the high doses to fully get rid of the cancer, and then replenish the blood system with a stem cell donation, which is obtained from a person, but when you get them from a bone marrow from another person, there are things like T lymphocytes and other aspects of the immune system that will actually attack hosts when donated. So if you can donate stem cell by itself, that would lead to the elimination of at least the donor attacking the host. And then getting beyond simply the donation of stem cells patient is the idea of combining stem cell therapy with gene therapy. If early attempts at gene therapy to create single gene defects involved the cloning of a gene into a plasmid, putting that plasmid or other genetic material into a virus, injecting the virus into the location of the disease, the virus would spread, in each case leaving a good copy of the gene. And in principle, that would work well. In practice, viruses generate immune responses, so there can be large immune responses to these viruses. You need to have viruses infect large numbers of cells, so it's hard to get a virus to infect enough of an organ to make the organ function well overall. And thirdly, there's certain cell types that don't divide much, like, say, for instance, neurons, where it's hard to get viruses to infect at all. So the opposite way of thinking about this is that if you were able to take an adult stem cell or a skin cell and use iPS technology, then you could put that single gene into that one cell and then grow the pluripotent stem cells with the corrected gene in it to however much you would need. You could have large amounts here, and then you could introduce that corrected stem cell into a tissue where over time it could populate large portions of the organ. Here you're taking one gene and trying to get it into millions or more cells. Here the gene only needs to get into one cell, and then you just need to grow that up, put that into the patient, and let that cell repopulate the tissue. So there are still risks with this approach. There have been some cases where the gene does happen to hop into a poor place in the genome, and leukemias have been the result of these types of gene therapy experiments. But this remains one of the most promising avenues. I'm sure that 20 years ago, people thought we would be there by now, and it's frustrating that we're not, but it is so potentially valuable that it's important that we keep exploring this in a very intensive way. Okay, thank you for listening.